Throughout life, it is understood that we as humans are exposed to a plethora of life-threatening dangers over the course of our lifetime. There are some that we can control, and there are others that we are never even aware of. In the modern era, these dangers can take the form of natural disasters, human-induced events, or even a pesky little microbe. This, of course, brings us to the star of this video, Bacillus anthracus. Known more commonly as anthrax, this microscopic organism has influenced our world in a multitude of ways. Dating back to the late 19th century, Robert Koch, a name many microbiologists might recognize, first used anthrax to develop his famous four Koch postulates. The microbe had been found in the blood of dead cows, and in his rigorous observations, Koch determined the four criteria needed to identify that a certain germ induces disease. In this case, Bacillus anthracus led to the betterment of humanity. Fast forward to September of 2001, and in the wake of one of the largest terrorist attacks on US soil, anthrax was at the root of yet another attack. Letters had been laced with anthrax and sent through the mail, leading to 5 deaths and 17 sickened, resulting in what the FBI calls the worst biological attack in US history. How exactly did a microscopic organism, once used as a spark for the study of modern microbiology, prove so volatile to humans? This question can only be answered through the understanding of what exactly is Bacillus anthracus. According to the U.S. National Library of Medicine, Bacillus anthracus is an aerobic gram-positive, spore-forming, rod-shaped bacterium. It is non-motile, and interestingly enough, the bacterium derives its name from the Greek word for coal, B. anthracus, because of its ability to cause black, coal-like cutaneous eschers. A fun little fact that gets a lot worse the more you think about it, let alone see it. Anyways, Bacillus anthracus grows optimally at temperatures of 37 degrees Celsius. It's pretty hot. And forms little white colonies on blood agar. The colonies do not form spores unless nutrients are exhausted and they're exposed to air. When these spores form, though, those suckers can survive in soil for decades. And boy, do they cause trouble. The problems associated with Bacillus anthracus stem from the toxins produced by the bacterial spores. Exposure to these anthrax toxins, depending on the infection pathway, are often fatal if not treated immediately. For the sake of this video, we will be discussing the immune response to the inhalation of Bacillus anthracus. Keep in mind that there also exists cutaneous infection and the less common gastrointestinal infection. The general explanation as to how Bacillus anthracus attacks the human body is this. Courtesy of UC San Diego Health, the anthrax bacteria can invade immune cells, called macrophages, and release potent toxins that paralyze key biochemical pathways, causing rapid cell death. Thanks for the gist, UC San Diego. Cool mascot, by the way. But what is really going on inside our bodies when experiencing an anthrax infection? Let's think of it like your typical Ocean's 11 big heist movie. When anthrax spores are inhaled, they lodge in the epithelial cells of the lungs lining, where they can persist for weeks or even months. Consider the bank infiltrated. Phase 2. Disable the security cameras. Bacillus anthracus runs into its first immune defense as it faces off against the innate immune system. Unfortunately for us, anthrax has a trick up its sleeve. There is a protein on the surface of the anthrax spores that recruits a regulator called factor H. This regulatory molecule is normally used as an inhibitor of the complement system. When functioning normally, the complement system is in charge of bolstering your body's defense of antibodies and phagocytic cells to clear microbes, promote inflammation, and attack the pathogen cell membrane. While this would be your body's normal response to a pathogen such as Bacillus anthracus, due to the uptake of the inhibitor factor H, anthrax is able to protect itself by inhibiting the activation of the complement system, subsequently crippling the innate immune response. Security cameras disabled. Phase 3. Knock out the guards. We're in the bank. Cameras are off. Now it's time to wreak some havoc. For an anthrax infection, havoc means tripartite anthrax toxin. This toxin is composed of a receptor-binding protective antigen, a metalloprotease lethal factor, and a calmodulin-dependent adenylate cyclase edema factor. The combination of protective antigen and lethal factor generates what's known as lethal toxin. These are the big guns. Lethal toxin goes straight for the security guards, or in the immune system's case, B cells. The anthrax protective antigen directly binds to two B cell surface receptors, tumor endothelial marker 8 and capillary morphogenesis gene 2. This bond is formed through the cation-dependent metal-ion-dependent adhesion site known as mitis, within its von Willebrand A domain. After the bond is formed, the protective antigen results in the lethal factor-dependent cleavage of mitogen-activated protein kinase kinases. This bond also inactivates inflammasome sensor protein NLRP1. While this attack is not strictly lethal to the B cells, anthrax lethal toxin exposure causes severe B cell dysfunction by reducing their proliferative responses to interleukin-4, anti-immunoglobulin M, and anti-CD40. The guards are knocked out, and the anthrax infection moves on with its plan. Phase 4. Cracking the vault defenses. There's only one thing standing between Bacillus anthracus and victory, and that is T cells. The only issue is, tripartite anthrax toxin isn't quite done yet. In addition to its components forming lethal toxin, the protective antigen in combination with the edema factor creates what is known as the edema toxin. T cells are already vulnerable to the anthrax infection as they express the tumor endothelial marker 8 and capillary morphogenesis gene 2, targeted in the last phase. However, their defenses are even further hindered by the edema toxin's ability to attack CD4+ T cells, preventing cytokine secretion of interleukin-2, tumor necrosis factor alpha, interferon gamma, and interleukin-5, as well as slowing cell proliferation. Edema toxin is able to slow T cell proliferation in a similar fashion to the lethal toxin by disrupting the chemical activation cascades. By raising intracellular C-amp levels, the edema toxin activates protein kinase alpha. This subsequently impairs RAS activation indirectly by activating the RAS antagonist RAP1. Under normal circumstances, RAS plays a critical role in the signal transduction of cell growth and differentiation for T cells, but this signal is now blocked by the edema toxin. The last safety measure is disabled, the vault is cracked, and unfortunately, In this hypothetical, the Bacillus anthracus has won. It is the champion. After all this explanation of how lethally effective Bacillus anthracus is at bypassing the body's immune system, it can leave you with an understandable sense of dread. There's not really much I can say to ease this feeling, but there is still hope. Inhalation anthrax is a serious threat, and when left untreated, only about 10 to 15% of patients survive. However, if caught early and combated with a rigorous course of antibiotics, about 55% of patients survive. There even exist some crude forms of anthrax vaccinations, widely implemented in the armed forces of countries like China and Russia. It isn't necessarily a death sentence, but this pesky little microbe is certainly a large enough threat that extensive research is warranted. I hope this video lesson taught you something and that you enjoyed. Thanks for watching. Here are some of my references.